Dear students, welcome and thank you for tuning in to this video. We are continuing our discussion about protein synthesis and structural material inside the biological organisms. And we talked in the previous video about the DNA and the human genome, which consists of 23 pairs of chromosomes with a total of 46. We'd like to look a little bit more deeper into these chromosomes to see what are they composed of, because this is important in understanding how genes are synthesized, and so we need to look at the structure of each of those chromosomes. So this is a schematic of a pair of chromosomes, and we call this short part as the short arm of the chromosome, and this is the long arm of the chromosome. We call this the centromere, which is the attachment between the two chromosomes. Remember that if we have pairs of chromosomes, this is going to happen after fertilization. So after your haploid cells have fertilized, the male and female, which is the sperms in the human and the eggs, and they will create this chromosome. Now each of those chromosomes is made actually of a long chain. If we want to break those chains, if this is a chromosome, if we go smaller, so this is called chromatin, and the chromatin actually is a chain of smaller chains. So we have our DNA. We know that it's a double helix strand of nucleotide, and if we have this double helix strand, we have another type of proteins called histone molecules. These histone molecules work like a disc that is going to allow the DNA to wrap around them. So usually we have 8 histones together to create like a ball shape, like this one, and the DNA gets rounded around it. So the DNA will be stranded around this collection of histone molecules. Now remember that histones are proteins, so they have to be created actually from cells. We're going to talk about this in a bit. Each histone, you have your DNA is rounded around it. So if you have 8 histones with the DNA wrapped around it, we call this unit as a nucleosome. So you have a DNA strand, you have histones. 8 histones will make like a ball-shaped spherical object. The DNA will be wrapped around it. The 8 histone and the DNA wrapped around it, they're going to create something called nucleosome. And the collection of nucleosomes, when they come together, they just organize themselves in such a way that they can create something called chromatin. And the chromatin is collection of chromatins will create our chromosome. So those are the structures of how the chromosomes are made. Now, in order to have proteins to create, so the DNA is carrying our genetic information, and that's why it's very protected. It's protected inside the nucleus, so there are no environmental changes that can affect it and destroy it or damage it. So this is our DNA. Now, in order to have it translated and transcribed and translated into proteins, there is intermediate steps along the way. The first step is that we need to unwrap this big collection of huge and complex structures, chromatins, in order to get to this the DNA double helix strand. And we need also to break the bonds between the double helix in order to get the genetic information. So it's a very complicated and delicate process in order to preserve our genetic information. That's important. So we're studying this in order to understand how proteins are made. So the first step was to understand what DNA is, which we know, and then what chromosomes are, and then go a step further to see how we are going to move our genetic information into proteins. I want to show you one more picture here. So this is electron microscope that shows on the top A, this is the chromatin. And the chromatin is what we said is the collection of all these nucleosomes. Together they're wrapped and organized in such a way that it will create this chain of all these molecules, which we call chromatins. So this is the chromatin, how it looked like, and it was isolated for experimentations, and it's 30 nanometers thick. And you can see from here, that's the 30 nanometer where it comes from. So these nucleosomes, which are composed of 8 histones as a rounded, spherical object, and the DNA wrapped around it, these nucleosomes, 3 of them, which is roughly 13 nanometers, will start creating our chromatin. Also, the B here, the other image, is showing you another electron micrograph. It shows the length of my chromatin, which has been experimentally unpacked. So you can see the nucleosomes here. After isolation, show the nucleosomes. So this is the chromatin, which is packed nucleosomes. Each one of these roundish objects is 8 histones, and around it we have the DNA wrapped on the DNA strand wrapped around it. So this is a presentation of the structure of the chromosomes and our DNA. The next step for us is going to be understanding how are we going to get the genetic information in order to be able to transcribe and translate it into functional protein. Thank you for watching. Until the next video. Bye.